So we have our sequences back from the sequencing center, and I've loaded them into this program called Genius. And what you see is there are two sequences for each sample, and that is because we sequence with two primers. We did the PCR with two primers, and we sequence with those same two primers. One is a forward primer. It sequences from the 5' towards the 3' end of the DNA, and the reverse primer sequences from the 3' to the 5', and so we get redundancy in our sequencing, so we can check to make sure in case there's a problem with one of them. So I've loaded them into the program. I've also loaded a reference sequence downloaded from GenBank. GenBank is this big online depository of sequences, and just about anyone who publishes using DNA sequences is required to upload their sequences to GenBank before publication. I've downloaded the Cambrian reference sequence for human whole mitogenome, and you can see the whole circular mitogenome, and it's been annotated. All the genes and control regions have been marked out. So let me show you what one of our sequences from one of the primers looks like. Okay. And what you see is a series of peaks, and these peaks are the readouts from the sequencing machine. So a green peak would correspond to a T, yellow would be G, red would be A, and blue is C. And so there's also base calls that have been made based on the actual output of the sequencer. So in this region, this is a very nice sequence. Single peaks that are real nice and clean, and the calls are colored in light blue to indicate high quality, and you can also see these bars above them are very high. That also indicates high quality. Now, darker shades of blue down here and lower bars reflect lower confidence in the base call, either because the peak isn't very high, or it's spread out, or potentially, see if I can find an example, there may be more than one peak in that area. This dark blue is very poor sequence, and you can see these peaks are long and there's lots of noise in there. Most of this sequence is very good, except for the ends, which is very common. It's just kind of an artifact of the sequencing process, that the ends are of low quality. So what I'll do is I will select our two sequences from sample 27, and then I'll also select our reference sequence. Okay, and then I will assemble these, and I will map our two sequences to the reference. Let me rename this. And alright, I'll open this up. Here's the reference sequence, and it's been annotated. Here's our sequence. It's just, 550 base pairs. We'll zoom in on this region. And our sequences have been aligned, and then have been aligned to the reference sequence. And then there's also this consensus sequence that has been made, which is a consensus of all three of the sequences, but weighted towards our sequences and not the reference. And then places where one of these sequences disagrees with the consensus, those base calls have been highlighted. So let's back up to the very beginning here. You can see the reference, lots of differences with one of the sequences. That's because this is very low quality. It's the very beginning of this read. We're gonna end up just getting rid of that. So what I'm going to do is I'm going to go through and I'm going to look for places where there is discrepancies between, well, really all three of the sequences and the consensus. Some places, like here, in this region, there's some disagreements with this one sequence and the consensus, but this is low quality reads right here. So as long as the consensus matches the good quality sequence read, then I'm not too worried about those. I am worried for places like this, where the consensus calls for a Y. That's an ambiguity code. And the reason for that, in this case, is because one of our sequences is calling a C and the other one is calling a T. The reference is also calling a T, but there will likely be mutations in our sequence compared to the reference. So I don't want to put too much weight on this, but looking at this, this looks like a T followed by another T. Up here, it's kind of a mess, but the T is the highest peak here. And I think this is a T, so I'm going to change this to a T call. Okay, now I'm going to look through the rest of the sequence and look for similar type ambiguity calls, or look for areas where there are differences, and make sure that the consensus agrees with the good read, or the better read, because this is bad quality, this is better quality. I trust the C call over the T, so just make sure that that consensus says C. Oh, this is interesting. So right here we have the reference is calling a T. Our two sequences are clearly C's. Let's zoom in. Those are clearly C calls. So our sequence is different from the reference. So this is a mutation. And it's these mutations that will allow us to assign a haplogroup to this sequence. So I'm going to just go through and check for any other interesting areas. This is an area of really nice sequence here. There's no differences. Oh, here's another mutation. The reference is a C, and the sequence is a T. Gonna go through. Okay, now we get to the end, and again, the ends are kind of ugly. I'm going to change this. Oh no, that's calling a C. That's, well, no, I'm not going to change that. But okay, so that's all there is to editing. Now what I'm going to do is I'm going to select this consensus sequence, the internal part, kind of trimming off the ends that I don't really trust because the sequence isn't of high quality. Oh, actually, I'm going to change this. That looks like an A to me. It should be an A. Okay. Now I'm going to select the consensus, except for the tail ends that are low quality. I'm going to copy that. Come over here to text file. This is actually going to be a FASTA file. I'm going to paste that in. And I'll include all the sequences. And so that's all there is to editing. I'm going to go back through and I'm going to edit the rest of the sequences and add them, save that, add them to this FASTA file, and that's what we're going to use to determine our haplogroups.